Hi, everyone. It's just about 4 o'clock, so I'm going to get us started by welcoming you to the MBL for this high school discovery program webinar. And what we hope to do today is just give you a little bit of a slice of life of our high school programs. And we have the faculty here to describe each one of their courses, as well as the rest of the education team. My name is Linda Hyman. I am the Director of Education here at the MBL. And we are really delighted to be able to be offering these programs a little bit virtually for those of you who have participated in some of our virtual visits. But also we hope in the not too distant future to go back to our hands-on, face-to-face, exciting program, which we'll share with you today. So normally Nipam Patel, who is our director and who is very involved in this program, and in fact is an instructor in it, would be joining us, but unfortunately he wasn't able to make it today, but you'll see some glimpses of him in the discussion of the courses. So I'm just going to turn it right over to Scott Bennett. And he's going to walk us through some of the logistics of the program. So Scott, take it away.
Hey, everyone. Let me just preface this while saying that everything that we're going to be presenting here is kind of in a non-COVID context. I know there have been some pretty encouraging news in the last few days, including this morning, about possible vaccines and things like that. But as of right now, we don't have a specific contingency plan for presenting these courses with social distancing and everything. But it is something that we are working on. But the way that these courses are going to be talked about and presented in this discussion is the way that they would normally be held in a normal in-person settings. Just keep that in mind while you're listening to these.
Although, right. Scott, can I just interject and say, however, we're really excited by the fact that we actually do have students here in COVID land.
True.
And we are actually operating a course, several courses actually. So it's not impossible. So hang in there with us. We'll be back to normal soon. Anyway, sorry to interrupt, Scott.
Yes, we want to make sure that to tell people that we do have the capacity to do it. So we are genuinely optimistic about these things. All right, so let me, all right, so the MBL's High School Science Discovery Program is a really unique opportunity for high school students who are interested in getting some experience with some really high level research and exposure to marine biology and their adjacent fields. And the High School Science Discovery Program is a fully immersive research experience. It's taught by the MBL faculty that you see here, who are leaders in their field. The program focuses primarily on hands-on research and getting involved in actually using the tools and techniques and implements that our researchers use to conduct their research. So students will get a chance to use cutting-edge microscopy and imaging tools, and there are also lectures and other didactic methods that the students will get experience with. Besides the on-campus portion of the courses, the students will also at their home institutions get pre-course materials, which will introduce them to a lot of the subject matter depending on which course they're taking, and kind of help them when they arrive on campus for their course to kind of hit the ground running, so there doesn't need to be any time taken up on campus working on certain things. And then when the students return to their home institutions, there will be, depending on which course that they take, follow-up materials that involve data analysis and other types of activities to kind of expand on what they learned and did at MBL during their course. So at the moment, we are offering 5 different courses. We have 4 7-day courses: Anatomy and Development of Marine Vertebrates and Invertebrates, Coral Reefs in a Changing World, Exploring Microbiomes of Marine Organisms with DNA Sequencing, and Adaptations and Evolution in Response to Environmental Challenges. And then a 3-day course, which is a CRISPR-Cas9 gene editing of aquatic and marine organisms course. So when the students are here on campus, they reside on campus and take their meals here. And this is a way to kind of enhance that fully immersive experience for the students. So what this allows them to do is to have breakfast and then go right to the lab and have as much time in the lab and exposure to the research that they're doing. A lot of the time, students will be able to have lunch with the faculty and other scientists who aren't necessarily teaching them their course. And so it's a great way for them to kind of enhance their experience by getting one-on-one time with other researchers. It also allows them the opportunity to kind of intermingle with each other. If there are students from other schools, they've got a chance to kind of interact with one another. And it also allows them to get as much experience as they can out of their stay by allowing them, say, after dinner, going back into the lab and either working on some things, finishing up, or conducting experiments that they have a particular interest in, if they want to kind of expand on what they're doing. I should go back just really briefly and say that the courses that are offered are typically offered during common school breaks during the winter, but we know all school breaks aren't the same, so we do have some flexibility. If there's any interest in the courses, we can work on the scheduling with individual schools or groups. So the Science Discovery Program is a cohort-based program, and that means that groups of students from different schools would attend and take part in these different programs, which is really nice. Sometimes we have one large group from a single school doing a course, and other times we've had smaller groups come together to take one course together, which is also really nice and advantageous for the students. At the moment, single students aren't permitted because of difficulties with chaperones. And we do require, for every 10 students that come from a school, we do require that there be one chaperone for every 10 students. In addition to the course-specific labs that the students will be doing and lectures, the students will also have the opportunity for extracurricular activities. And so shown here are some of the places that the students will be able to kind of tour and get a firsthand experience with. So we've got images of our Marine Resources Center, where we hold and maintain all of our locally caught marine organisms that researchers and external researchers use for their work. They'll also get a chance to take a look at our cephalopod initiative, which is a culturing program right now dedicated to kind of optimizing the rearing and culturing of different cephalopods as model organisms, and also get an opportunity to tour our MBL Hui library, which is a really interesting place, and they'll get to see the rare book collection and also do things like handling a Nobel Prize that we have on hand. And then there are other activities that students can do in downtown Falmouth. Granted, the winter isn't exactly a bustling time of year, but there still are options for students if they want to go off campus with their chaperone to do certain things at night in their free time. So the tuition for the week-long courses is $2,500 per student. And the 3-day course is $1,250 per student. So this tuition cost includes all the housing accommodations, meals, as well as the lab supplies and equipment, and includes the hands-on lab and classroom instruction from our faculty and a lot of their lab associates and the people that work with them. So, but also through a very generous gift, the High School Science Discovery Program is able to offer scholarships for some public high school students. And if this is an option, something that we'd be more than happy to discuss individually with people. If you have questions about that, you can feel free to reach out to myself or to Jean or Linda, and we'd be happy to discuss those situations with you. So that's kind of the background, the general background. And again, at the end, if you have questions about anything you hear during the individual presentations, we'll have a very quick Q&A session where you can ask questions of the different courses or other follow-ups that we'll be happy to answer as they come up. So let me turn it back to Linda.
Great.
And we can get started with the individual course presentations.
Fantastic. Thank you, Scott. Yeah, so we have several of our MBL faculty here to describe the courses that they've been working on. And so I'm going to introduce Lisa Abo first, who is a director of our anatomy course. She'll tell you all about herself. She's also our veterinarian, and we're delighted to have her participate. So Lisa.
Okay. Thank you, Linda. So my name is Lisa Abo. I'm the veterinarian at the Marine Biological Lab, and I'm one of the instructors for the Anatomy and Development course for marine vertebrates and invertebrates. So the objective of this course is really to expose students who have an interest in biology and just science in general to a range of topics that appeal to their innate curiosity. So we accomplish this by using a comparative approach to anatomy with a variety of marine fish and invertebrates. And then we use that foundation to discuss developmental biology. So when creating the lectures for the course, I felt it was important to get the students into the lab as much as possible. So for each lecture, there is a corresponding lab. So the course consists of 2 to 3 hours of didactic learning each day and an additional 4 to 5 hours of hands-on laboratory experience and tours. Sorry. Okay, so we begin the course with an orientation to the equipment that the students will be using throughout. And we're able to provide one microscope for every 1 to 2 students, depending on how many students are in attendance. So there's never a time when they don't have access to the equipment. Throughout the course, they will learn how to care for marine fish and invertebrates, and they are tasked to set up their own tanks. They select animals from the Marine Resources Center tank room, and then they have to monitor their water quality every day and take care of the animals. The students will work in teams of 2 and dissect a teleost, an elasmobranch, and a cuttlefish. And then there will also be dissection demonstrations of other invertebrates, so sea star, urchin, whelk, and clam, and kind of what is available to us at that time. And then some of the other lectures will be on marine biodiversity, coral biology, cephalopod biology, and then those will all have corresponding labs and tours. There's a lecture and corresponding lab on diagnostic procedures, where the students will administer anesthesia to a fish. They'll learn about the biology of horseshoe crabs, and they'll even collect a blood sample for a microscopic exam. And so the full immersion activities really enhances their learning, and then it also fosters an enthusiasm for science that we hope will last throughout their lives. The program gives students access to very specialized equipment, and then it also gives them a unique opportunity to see what scientists and aquatic veterinarians like myself actually do. And it gives them a chance to work in the same labs and use the same equipment that scientists at MBL use. The teachers are also encouraged to participate and really seem to enjoy themselves. So now I'm going to hand it over to Nipam Patel, who will talk about the developmental biology section of this course.
Hi, everyone. Thanks. Let me just get my screen sharing going here. So as Lisa said, I also help to teach the developmental biology section of the anatomy and development course. So, Lisa talked about the fact that the students get an opportunity to look and dissect quite a number of different species of fish and other marine invertebrates. And what I do when I lecture about developmental biology is try to describe to the students how those organs and tissues come to be, how the cells differentiate to that fate, and how they get positioned properly in the embryo. And so this is kind of the key to the whole topic of developmental biology, how you go from a single cell into a multicellular organism that has the right cell types and the right organization. And so this is what we go through with the students, and we describe both a little bit of review of biochemical and genetic processes that go into this. But for me, the real joy of the course is the hands-on aspects in the lab. So as Lisa said, we try to get the students into the lab right away and to have them spend as much time as possible in the lab learning. Lisa mentioned that each student or pair of students has their own scope that they work with. Not only is it a scope, but it's actually a really well set up system with a camera that can record images and movies. And so this is something that we use actually in quite a lot of the courses that we teach at the MBL. But it's a great opportunity for the students to be able to look at things and record and make observations. And what I focus on is for them to use it to understand a bit about developmental biology. And we take advantage of quite a number of systems that we can show the students. So one of them, for example, is zebrafish. So they get a chance to look at the anatomy of fish. And what I do with them is actually get them to watch zebrafish development and talk about how things like the nervous system and the muscles form. So this is a movie taken by one of the secondary school students last year. And it's just a really nice overnight movie of zebrafish development. So you can see it going from just a flat sheet of cells into this thing, which obviously you can see the eyes and the repeating pattern in the back and everything. And they get to watch these and they get to watch them hatch and everything and record and understand what they're doing. The other thing we get to do with them is to really allow them to use some really advanced microscopes to do some analysis. And so this is one of the confocal microscopes that they use. So it's always a lot of fun. We explain to the student that the microscope is worth roughly $500,000 and that they should try not to break it. But honestly, we tell them the whole point is for them to learn to use it. And we sit there and really explain to them how to use it and get them to collect their own data. And it's really a lot of fun and to get to watch the students do this. And then we also teach them some basic software that they can load onto their computer, take home with them, that allows them to analyze the data. So again, these are 3 movies taken by 3 students last year. And on the left is a zebrafish, in the middle is a Drosophila melanogaster fruit fly embryo, and on the right is a cuttlefish. And you can see that the students have stained for muscle and nervous system and then created these 3-dimensional reconstructions of the data. And so this is something that they get to do during the course, and they get to take the movies home with them and post them to social media and everything. And but more importantly, they learn how embryos develop and how we get organ systems and things like that. So it's a great experience, I think. And finally, we also do some stuff where we, sorry about the music, where we have them image butterfly wings as another very cool way to learn about microscopes. So with that, I'll go ahead and end, and I'll pass it on to Loretta Roberson.
Great, thank you, Nipam.
I will stop my sharing. There we go.
Oops. Okay, so yes, I'm Loretta Roberson. I'm an associate scientist here at the MBL. And my research focuses on corals and how they're able to build these, understanding how they're able to build these fantastic structures that you can actually see from space, and the role played by the symbiotic algae that live inside their tissues. So I lead the Coral Reefs in a Changing World course, and I want the students to get a better understanding of corals and reef biology and ecology at both macro and micro scales. So we start the course by first learning about just why corals are important for both the reef organisms and humans, and get a better understanding of the rich diversity and beauty that reefs have. Then we dive into more hands-on exercises in both the field and the lab. And we study coral biology, but things that can be used as just basic biological principles as well. So for example, we talk about how coral reefs in the Caribbean alone generate almost $9 billion a year in revenues based on coral reefs. And in Pacific Islands, people can receive up to 90% of the protein from coral reefs themselves. I also include activities that make use of the rich online data sources that we have for marine environments. And these can be accessed by teachers and students, both pre and post course. But what I do is I show the students how to access these sites, how they can download and manipulate the data, and then also show them how they can analyze that data, make graphs in Excel. These can also be customized, depending on the student or course or teacher interest. And it can be either, something of personal interest, a hometown, or around specific events. And so like the graph you see here was when Hurricane Sally came ashore this fall. Now, working in the underwater environment requires special skills and tools. And so I take the students through all the different techniques that we use to study corals, and in so doing, take advantage of our close collaboration and relationship with the Woods Hole Oceanographic Institute. And the students in the course will be able to themselves operate, sorry about that. They will be able to operate a remotely operated vehicle on their own and explore the environments around Woods Hole. And last course, a student was quite surprised to find a harbor seal checking them out during their dive. It's one of the nice things about working out in the field is you never know what you're going to see. We also learn about really important contemporary issues related to both corals and general science. And while we don't have CRISPR-Cas9 working in our coral system yet, the students will learn the basic techniques and tools that are used in those types of studies. So microinjectors and micromanipulators, but in the case of the coral course, it's within the context of the global decline of corals and coral bleaching. They touch on conservation and how we might be able to help corals through rescuing them by reinfecting or injecting them with their algal symbionts. But as Nipam said, a lot of the activities in the course revolve around making use of all the incredible resources we have here at the MBL, and including techniques and equipment that were developed here. And so again, as Nipam said, each student will be able to use these equipment themselves and learn how to use it and how to manipulate the images afterwards. So in my course, we focus mainly on bright field and fluorescence microscopy, confocal laser scanning microscopy, and scanning electron microscopy, or SEM. And all the images that you see here were taken by past students in the course, and they were really excited and like I am when I see these organisms at different scales and perspectives. It's really inspiring, and I feel like the students get inspired as well. So thank you for your time, and I'll pass it off then to Dr. Hillary Morrison.
Hello, I'm Hillary Morrison, a senior scientist at the MBL. I primarily study the microbiomes of host organisms such as macroalgae. I also oversee the MBL's sequencing facility and co-teach the Marine Animal Microbiome course with Director of Research David Mark Welch. We designed a course to introduce students to this emerging field, to the truly profound realization that's come about in the last couple of years. That is, that an organism is not just composed of its own cells. It can contain a far greater number of cells of microbial origin, the so-called microbiome. The microbiome influences an organism's health and well-being. There's been a tremendous amount of work done on the human microbiome. We know a lot about the effects of resident microbes on behavior, metabolism, and pathogenesis in humans. This course won't look at humans. We're a marine research laboratory after all. Instead, the students will have the chance to sample and study the microbiomes from a wide variety of marine organisms from the local waters that are available through our Marine Resources Center. Like the other course offerings, we teach through a combination of lectures, short exercises, and labs, where the students will work with animals and carry out molecular biology procedures, such as DNA extraction and PCR. Students will spend a significant amount of time on bioinformatics, which is what drives this field. The students will learn to do the sorts of computational analyses that microbial ecologists use today. Of course, we want to get the students out doing things as much as possible. Here are some of the students from previous courses examining organisms in our MRC. They learn about them from experts like Scott Bennett and Dave Remsen. They handle the animals, explore what's known about them, and decide which ones they'd like to study more. A critical component of the course is that the students design their own experiments within certain constraints. They decide what organism to study and what variables or conditions to examine to address a question of interest to them. Students can work in pairs or small teams. They can combine sampling efforts to have greater access to data sets. On day 1 or 2, they'll collect samples from the organisms. The least invasive sampling is of an animal surface, but they may choose to investigate other sites, perhaps in conjunction with the anatomy course. In later lab sessions, they extract the sample DNA, do the polymerase chain reaction amplifications, and have short sessions during the downtime for microscopy, additional field trips, or MBL tours. Their DNA samples will be sequenced, but the entire process takes longer than the 1-week course. So while they're here doing lab work, they'll be learning bioinformatics using data generated previously by earlier sessions of the course, by MBL scientists, by summer scientists, etc. After the course ends, we will deliver their sequence data sets to them, and they can analyze them back at their home institutions. So they'll be able to bring what they learned back to their secondary schools and continue what they started at the MBL. There's no better way to demonstrate this than to show you a student presentation, not polished, but quite enthusiastic. These 3 students, Alana, Emily, and Morgan, examined sea urchins, moon snails, and whelks. They did some online research to learn a bit about each. What is it? What does it eat? Who eats it? Why is it important to biology? What is known, if anything, about its microbiome? Usually that's very little, maybe a paper or 2. These students will be examining most of the organism's microbiomes for the very first time. These 3 students looked at 2 animals of each type and, as a control, the water from the tank in which the animal was housed. This is how the students formed their hypothesis. Is the microbiome of an animal reflective of the water they live in, or is it different? Has there been selection? Are the microbiomes of each species different from each other? Is it random, or do the microbiomes of the members of the same species show consistency? Here's an example of the kinds of things they looked at. These bar graphs are common in microbial ecology and microbiome studies. Each color represents the relative proportion of a different kind of bacterium found on the surface of the animal. They can use their laptops to go to NCBI or other databases and look up these types of bacteria to see why they might be important. This is a relatively sophisticated statistical analysis. Principal component analysis. It's a way of separating the data sets from one from another. You can see that the students found the microbiomes of a pair of whelks to be very similar, a pair of moon snails to be very similar, and those from a pair of sea urchins to be very similar. The dots are almost overlapping in some of these cases. However, the microbiomes of whelks, snails, and urchins are very different from each other, and they are different from the tank water in which each of the organisms were housed. So these students, in 1 week's time, were able to show that surface microbiomes are unique and have adapted to be the microbiomes of particular species. As the students do this more and more, they may collectively generate enough data to publish a paper. So we think this is a very exciting course. The students get to do a lot of hands-on work as well as informatics, which is one of the fundamentals of biological research. So I'll end here and introduce next Dr. Karen Echeverry to tell you about her course.
Thanks, Hillary. Okay, so I'm Karen Echeverry, and I'm the course director for the CRISPR-Cas9 gene editing course of aquatic and marine organisms. So I want to start first by just telling you a little bit about what CRISPR-Cas9 is. So CRISPR-Cas9 is really a revolutionary technology which allows you to rewrite the genetic code in a cell. And so the Nobel Prize in Chemistry was awarded this year for the discovery of this technology. And so essentially what it means is that by understanding the genetic code, we can actually go in and we can use this technology to cut out a segment of DNA or to actually insert a segment of DNA. And what this does allow us to do in terms of biology, so we can think of this really like a Swiss Army knife. It's not just one thing we can do in a cell, but we can use it to knock out a gene to understand the function of that gene in development. We can rewrite small portions of the genetic code, for example, to change a mutation which may be causing a disease. Or as in basic research we often do, you can use the technology to insert a new piece of DNA to allow us to follow a specific gene in a cell. So what can high school students do with this technology? So as you've heard earlier, zebrafish is a commonly used research organism. And one of the reasons that it's used is that it develops very quickly. So what you're looking at in these images here is a single cell embryo. And within 24 hours, you can see it's a fully developed swimming fish, which has all its organs developed. You can see the eye here, the heart is here. It has a full vasculature and nerve system. And so the genome is sequenced of this animal, which means we can manipulate its genome. So in this course, students are going to get hands-on work, actually learning how to inject the constructs which will allow us to knock out a gene using this CRISPR-Cas9 technology. They're going to learn microscopy and how to identify phenotypes. So what does this look like in a lab? Actually, here's a student here sitting at a dissecting scope. And what you don't see here is it has a manipulator holding this very fine needle. And we will give the students pre-designed what we call guide RNAs with the Cas9 protein, which will cut a specific piece of the genome. And they're going to inject this complex with the dyes that are able to see what they're injecting into the single cell embryo. And then we're going to use microscopy and look at what's happening to these embryos over time when they've knocked out a specific gene. And this is an example of what they will actually do in the course. So what you see here is 48 hours later is a normal developed zebrafish embryo with all the pigment along the body axis. But here where they've knocked out this gene called SMUDEN, you can see that the animal doesn't develop normally, that it has a bigger heart area and its body axis has not developed normally. So this is just one example of what the students will do in the course. And as I said, this is a widely used organism for research. So we also have what we call transgenic lines where fluorescent proteins have been inserted where you can label, for example, the nervous system or the vasculature, and the students will also get to use these transgenic animals and inject these CRISPR-Cas9 constructs to knock out genes which may affect the development of these structures. So there's going to be a big emphasis on hands-on work in the research lab, but every day they will also have lectures which will teach them the background in molecular biology of this technology. As we're using zebrafish embryos, they will learn more about developmental biology and microscopy, like what Nipam talked about earlier. There is obviously great power with this technology, so we will have discussions in class on its relevance to human health. And this is in many ways. So it's not just an animal cell. You can also manipulate the genome, for example, of a plant cell, and we'll discuss how this may affect the food chain. We'll also discuss its implications for treating human disease. And of course, with such powerful technology, we also need to discuss the ethics of this use, especially in humans. The students, while they're here for this 3-day course, will also introduce them to other research organisms which we use. These are 2 I use in my lab. So this is a local sea anemone, and this is a type of salamander called an axolotl, which can regenerate multiple body parts. And CRISPR-Cas9 technology can be used in both of these research organisms too. And then we'll also give them tours of the various facilities, especially showing them how you actually culture a large number of these animals. So that's my course in a nutshell. And now I'm going to pass you over to my colleague, Kirsten, for her to tell you about her course.
Great, thanks, Karen. Right, so I'll be leading the course on adaptation and evolution, in which we use examples from aquatic biology. So in this class, students will really explore some of the fundamentals of evolutionary biology, which includes concepts of adaptation, fitness, and genetic heritability. We'll talk about the role of life history strategy and organisms' abilities to change their life history strategies in response to environmental conditions, and how that determines fitness and allows adaptation. We'll talk a lot about environmental stressors and environmental change and the ways in which organisms may or may not be able to respond to those stressors and changes. So this course will really be a mix of activities, including week-long projects, short lab experiments, lectures. So we'll use rotifers as the main model organism for this class. Rotifers are aquatic microscopic invertebrate animals. They're found nearly every place that you can find water. They're really diverse. They've evolved to and adapted to handle a wide range of environmental conditions, and they're really important components of aquatic food webs. So because of these reasons, and because rotifers are really easy to use in the lab, they're easy to grow, they have fascinating behaviors that are easy to observe under the microscope, and they have really interesting life cycles. They make a great study system for this class. And for those same reasons, rotifers are really easy to use in the high school classroom when you go. So my show is advancing on its own, I'm not sure why. They're really easy to use in the classroom when you get back to your own school. And the class will include plenty of other examples of diverse aquatic organisms, really ranging from bacteria to whales as we move through the class. So in the lab, students will use a variety of techniques, like in all the courses. This includes culturing the organisms, pipetting, microscopy, molecular biology, data analysis, and bioinformatics. So for example, we'll take advantage of some of the unique traits of rotifers, such as their tendency to swim toward a light source, to conduct really simple but powerful experiments that demonstrate their ability to adapt to environmental conditions. Students will, from these experiments, generate data, analyze and graph the data, and discuss the conclusions that they can draw from their experimental results. We'll also do some simple sampling from local aquatic environments, examine those samples under the microscope, and then use some basic molecular biology techniques, including DNA extraction, PCR, gel electrophoresis, DNA sequencing, and bioinformatics to demonstrate evolution at the DNA level and to discuss evolutionary relationships between diverse organisms. And then finally, we'll learn how studying
these traits and mechanisms in model organisms, a wide range of model organisms, can actually help us to understand human biology. Students will, I hope, understand that it's the evolutionary relatedness between organisms that allows us to do research on a variety of plants and animals and cells, and how that teaches us about fundamental biological processes that are relevant to people. So I think this will be a fun and exciting, engaging course for students. And by the end of the course, students really will have gained exposure to some of the real central questions and theories in biology, and will have participated in the research process to address these questions. With that, I will hand it back to Linda.
Well, thank you. Thank you all. I hope this is a little snapshot of the courses that we have to offer, the diversity of experiences here at the MBL, but also what you can hopefully see that they all share in common is not just the love of science, but the access to the instrumentation and the state-of-the-art technologies and the fabulous faculty we have here that are just dedicated to working with high school students to share that sense of excitement. So just very quickly, I'd like to next introduce Jean Enright, who is our Director of Student Affairs and manages the high school discovery programs and the person who arranged this evening tonight. And she's going to lead a question and answer session in case there are any questions out there. So Jean.
Thanks, Linda. Welcome, everybody. We'd love for you to unmute yourselves and turn your video on so we can see you folks. But we'd like to open it to questions now. And you can either ask your question or you can go ahead and put it into the chat as well.
And don't be shy. Amy, I know you have questions.
You mentioned the cost for the students. Is it the same cost for the faculty that travels with them?
Jean, you can take it or?
The faculty's room and board is covered, actually. So, and we do ask for, one chaperone, teacher chaperone per 10 students.
Is there a limit amount of the amount of students that can come from any given school at a given time?
Not really. It depends on the capacity that each course can accommodate, but we did have several students from one school last year. We had about 20 students from one school last year.
And can students attend, like if you have one group come, can some students go to one course and one students go to another course?
Absolutely,
yeah.
yes.
Okay, cool, thank you very much.
I have a question. My name is Courtney. I am a parent, so I don't know if this was really directed more towards teachers, but I'm familiar with the MBL. So just wondering how I can get my student who's a freshman involved. I don't know if it's at his school. How can I find out how to get him potentially enrolled in one of these programs?
Linda?
Sure. So, the point of this webinar is just to introduce the concept to really the whole community. So what we would urge you to do, and we'll be happy to facilitate this, is to have a conversation with the administration in the school and see how we could work together to pull together a group. So again, as I think Scott mentioned, we're at this point in time really basing this on a cohort model. So rather than have individuals coming from separate schools, to have folks travel together with a teacher and create that kind of community. But it's got to start somewhere, and no better place to start with than a parent. So we're delighted you're here, and it's absolutely fine that you're here. We're excited to have parents here that share the excitement of what the MBL can bring to our students. So happy to help you navigate that, but that would be the method.
Okay, because he goes to Boston Latin School. So I don't know if you've had a group working through Boston Latin School.
Not yet, but it's a great school. We'd love to have you.
Okay, well, and I'm familiar with the MBL because my grandfather did all of his research, lifelong research there. So
Wow, fantastic. Wow, that gives me goosebumps. That's
no,
wonderful.
and we're still in Woods Hole. So
That's
we're,
great.
you know, anyway, we still have very deep ties. So so I'll find out from Boston Latin and see if there's anything that can be done.
Yeah, and we'll be happy to work with them. So just send them our way.
Thank you.
Okay, we do have a question through the chat. Is this program just for groups in Massachusetts? No, it is not. We've had students from schools in Texas, Illinois, New Hampshire, Connecticut. So it's open, absolutely.
What's the timing as far as signing up? So I'm looking at probably 2022. When should I engage in the like, okay, let's go, let's make this happen?
I'd say if we're, and Linda, correct me if I'm wrong, but if we're thinking of spring, say of 2022, you'd want to start, let's start discussing in the fall of 2021.
Again, this is ideally set, and not necessarily, but ideally set to correspond with vacation times, so that, you know, again, that students can come freely and not have to worry about their curriculum at home and that sort of thing. It's also a great place to spend a week on your vacation. So depending on your timing, we can, you know, work with you on that.
So is the 2021 program still happening?
Oh, isn't that the $1 million question? I remain optimistic. It's, you know, it's unlikely that we'll be able to support a full-fledged residential program, although we do have students here now who are, you know, in residence, and they're doing great, and we're delighted to have them. Being realistic, you know, we'd be happy to entertain such an idea, but it would really have to depend on many, many different things that have to fall in place. Now, if you happen to be a local school and are able to, you know, come for the day, we can work with you. Where are you from?
We're in California.
Yeah. Yeah. Well, California is a compliance state, so that's a good thing with regard to COVID isolation procedures, et cetera, here in Massachusetts. But it has obvious complexities this year. But again, I mean, I'd be happy to chat with you offline and, you know, see if there are any opportunities that we could explore together.
Sure, that'd be great.
Any questions for our faculty?
I guess really quick. My son is a sophomore this year, and he was watching part of the presentation, and he was sort of shocked by everything that he would be exposed to. And he kept telling me, I don't know how to do any of that. Am I going to be ready for a class like this? And how long do I have to learn all of this stuff? And I said a week. And so what are the expectations for students before they come? He has 1 year of biology under his belt and love for marine animals and mammals all his life. So
that's a good start. I'm gonna ask the faculty to chime in on that because each of their courses is a little different, but who wants to comment? I know we've had students that were freshmen to seniors, so it definitely runs the gamut. Loretta, you're muted.
It didn't unmute me. Okay. Yeah, I can say something about that because in the courses I've taught, we had actually, you know, 3 different levels of students in the course. And so the seniors were, you know, much far ahead and more advanced and could get more out of a lot of the activities. But that said, even the freshmen were taking part and learning, you know, at their own level. And at least I think it's important that they just get their hands on it, and just going through the experience is really helpful and empowering for some of those, you know, students that are feeling more timid about what they're being exposed to.
So I'll add that this is where the relationship between the teachers at the home institution and MBL really comes into play, because the cohorts that are selected, at least so far, have been selected by science staff. And so they have a very good understanding of where their students are and who might be good for the program. But the really nice thing, and I've said this before about our program, is that although it's really high level science that these students are being exposed to, it's taught by our amazing science staff and faculty in a way that's really accessible. And once I've seen a lot of these labs happen, a lot of these courses happen, and once the students actually get their hands on microscope and start looking at these things and start just being exposed to them, they realize how uncomplicated a lot of these kind of concepts are. And so that helps incredibly. And the other thing that's really important is that the pre-course kind of instruction that the students will take part in before they arrive. And so with the pre-course material, they're able to our faculty kind of introduce them to a lot of topics that they might not have seen before, a lot of the content they're going to be dealing with, so that when they arrive, they can kind of hit the ground running and they don't have to learn new things. They can do it in the comfort of their own home institution before they arrive. So that's.
And I'll add one other thing too, that Nipam had mentioned this, and you too, Scott, that, you know, post-course too, they're bringing a lot of the material that they gather at home with them. And we work with the faculty to help them access those files and analyze them, et cetera. And they continue on their own and learning more. So.
In the case of my course, it's very easy to get their data to them, and all they'll really need is computer access later on. And I did not explain during the recorded presentation what VAMPS was that was on the student presentations in great excitement. But it's our basically it's a server where you can analyze microbial data. It stands for Visualization and Analysis of Microbial Populations. And so students can set up an account and use that after they go home. There's no problem with that at all.
Well, if you have other questions, you know where to find us. And we just thought, you know, to end with not hearing from us, but rather hearing from the students that participated in the course. And this has been on our website, so I apologize if you actually had a sneak peek at it before, but I'm going to ask Scott to cue it up, and it'll end our presentation for this evening. And at the very end, of course, there's our website and ways to contact us, so please feel free. And I'm really looking forward to a normal year in the not too distant future, where we can really welcome you all here with the students, especially. We're kind of lonely here now. It'd be nice to see you here. So, and with that, Scott, thank you all. Thank you, faculty. You are amazing. And really appreciate you being here with us this evening. And Scott, why don't you roll it, and we'll say good evening to everybody else.
All right.
I attended an MBL course on the anatomy and development of vertebrates and invertebrates. It was an experience that I will never forget. It was the opportunity of a lifetime to meet amazing researchers who taught me to appreciate the interconnectedness of the various branches of science and research.
I attended a session at the MBL during my senior year, and it was truly one of the most memorable and impactful experiences I've ever had. The MBL gave my peers and me access to all of their resources, allowing a curiosity and passion for science and discovery to thrive. My favorite part of my time at MBL was the incredible lab experiences offered. I dissected vertebrates and invertebrates, observed zebrafish embryo development under a microscope, anesthetized a fish, and even got to image a cuttlefish embryo using a confocal microscope.
For me, the most enjoyable aspect of MBL was learning. My experience was very different from the science experiences I've had in school because it was much more hands-on. It felt more professional. Everyone there is so nice and so excited to teach you.
MBL is significantly more hands-on, which makes it a more fun and memorable experience. I'd much rather be pinched by a horseshoe crab and have someone talk about them instead of just the talking portion. But you also get to handle a wide variety of living specimens that you can hold or put underneath a microscope. The location and the people who work at MBL will make it a truly unique experience.
The location of MBL allowed for the lab experiences to be really unique. Many of the animals are from the local area, making me feel immersed in the local culture and community.
Yes, I would definitely recommend these courses to other students. Even if you have a smidgen of interest in science, you will have a blast.
I would definitely recommend MBL to other students, and I have.
I would definitely recommend the MBL Science Discovery courses to other students. Even if a student isn't planning on pursuing science as a career, the unique and diverse experiences at MBL will push you outside of your comfort zone and challenge you to become a better student.
I think one of the most important things that you should know before you go to MBL is just to ask a lot of questions.
To challenge yourself. Ask questions, engage directly during the labs, and allow your curiosity to flourish.
Don't be afraid to ask questions and explore. If you see something interesting, put underneath a microscope and look at it. It's what you're there for.
To students interested in attending a course at the MBL, my advice is to remember that a challenge is always just another reason to try harder. The MBL will challenge you, and it's important to know that asking questions and being curious and pushing the boundaries of your knowledge are the marks of a great student and an innovative thinker.
I would come back to MBL for another course in a heartbeat.
I'd love to come back to the MBL for another course.
And I would definitely come back to MBL for another course.
Would I come back for another course? Absolutely, without a doubt, yes.
The High School Science Discovery program currently offers 5 courses: Coral Reefs in a Changing World, Anatomy and Development of Marine Vertebrates and Invertebrates, Exploring Microbiomes of Marine Organisms with DNA Sequencing, CRISPR-Cas9 Gene Editing of Aquatic and Marine Organisms, and a newly developed course, Adaptation and Evolution in Aquatic Animals, featuring the fascinating aquatic organism, the rotifer. For more information on courses, please visit mbl.edu/education and click on High School Science Discovery Program listing.
Thanks, everyone. Please be in touch. And it was really lovely to see you all this evening. And again, thanks to the faculty and everyone, Jean, Nipam, Scott, and be in touch. Have a good evening, everyone.
Night.